G'day everyone, I'm Michelle Power, a parasitologist from Sydney, Australia. I'm coming to you from a park that's not very far from the city centre. It's about 2 kilometres from the Sydney Harbour. Today, myself and a number of colleagues, other parasitologists and students will tell you about our favourite marsupial and also their parasites. And just to let you know that we are still in lockdown, so we'll be zooming in from our homes and various sorts of places, or some others might have been able to get out into a park like I have. While I am out in a park, we won't see any marsupials. There's a few marsupials that have made cities their homes and can be found dwelling in our rooftops of houses, but marsupials are nocturnal, so the best time to see them and hear them is during the early morning hours or the late evening hours, so at dawn and dusk, or during the night when you're spotlighting. But what we can do is we can find remnants of marsupials and we can go and look on the ground behind us, and we'll be able to locate and collect and find exactly what a parasitologist needs to do their work on parasites. So we're here to talk about parasites in animals with pouches, or marsupials. So there's about 330 marsupial species around the world. So they occur in South America. There's one species in North America, but most species are found here in Australia. And we have around 220 marsupial species. So the ones you'll be familiar with will be the koala and the kangaroo. But we also have other species that have adapted to different environments in Australia. So for example, tree dwellers. So we have the sugar glider here that you can see on the slide. Sugar gliders have a membrane that runs from their forelimb to their hind limbs and allows them to glide between trees. So it's not flying, but they can glide. They're not like wings, okay? We have carnivorous marsupials, and pictured in the slide are quolls and Tasmanian devils, and you'll hear more about Tasmanian devil parasites a bit later on. And also Antechinus, which is a little insectivorous marsupial that looks very much like a mouse. So I mentioned that you have in North America, a marsupial species called an opossum, pictured here, so it's a carnivore as well. And so you can see here that the opossum is carrying its babies on its back instead of in a pouch. Now, some marsupial species do this. After a certain age, the babies get too big to be carried in the pouch, and so they're carried around on the back. Another example is the koala, pictured here, and also Tasmanian devils will carry their offspring that way as well. And of course, though, kangaroos, which you see here, are carried around in the pouch for quite a long time. And you'll often see very big kangaroos that can, baby kangaroos still, but maybe like teenage kangaroos, still trying to get their head back in the pouch and climb in, but they're too big to get in there. And that's a behaviour that we see often when we're out working with kangaroos in the field. So the parasites that we find in animals with pouches, or marsupials, are the same taxonomic group as parasites that we find in some of the animals that you would have heard about in the other sessions of this parasite festival. So ectoparasites, fleas and ticks, and endoparasites, worms. But we also, today, will probably focus more on, on my favourite parasites, and they're the protozoa. So the 3 parasites that I work on, or that I'm interested in, they're so small that you actually need a microscope to be able to view them. And so here they are, pictured on the slide. So that's Cryptosporidium, Giardia, and Dimeria. But before I talk about my favourite parasites in probably one of my favourite animals, the Tasmanian devil, we're going to hear from a number of my colleagues talking about their favourite marsupials and also their favourite parasites that are found in or on marsupials.
Hi, I'm Manushika Herat. I'm a postdoctoral researcher affiliated to University of Sydney and Macquarie University. My favourite marsupial is the common brush-tailed possum. Those who have seen a possum knows that they have the cutest pink nose that you have ever seen. Brush-tailed possums are native to Australia and distributed throughout the continent in both urban and natural landscapes. They are also found in New Zealand, where they are considered as a pest. Brush-tailed possums host a lot of parasites. However, my focus today is the protozoan genus Cryptosporidium. Cryptosporidium can cause enteric diseases in a range of vertebrate hosts, including humans, and also can be fatal for immunocompromised individuals. There are nearly 38 species of Cryptosporidium species and 40 genotypes identified so far. Most of these Cryptosporidium species or genotypes are host specific, that is, they cause diseases in particular hosts. However, some Cryptosporidium species can cause diseases in multiple type of species as well. The Cryptosporidium genotype found in brush-tailed possums is unique to them. Because of that, this host specificity, it is less likely for brush-tailed possums to spread Cryptosporidium to other species. However, Cryptosporidium species or genotypes, there are two other genotypes found in brush-tailed possums, which is somewhat similar to Cryptosporidium hominis and Cryptosporidium parvum, which is a major cause of cryptosporidiasis in humans. But all these three genotypes found in brush-tailed possums are genetically and morphologically different from any other identified species or genotype. Because of this reason, it is less likely for possums to spread diseases to other animals. Currently, we are looking at and studying urban adapted brush-tailed possums to see whether they host Cryptosporidium parvum and Cryptosporidium hominis as a result of urban adapted possums using anthropogenic resources and the likelihood of reverse zoonosis from humans to animals through possums. Thank you.
Hi everyone, my name's Tanya Suki from Melbourne University, Australia. My favourite parasite in marsupial? It was a tricky question because there are so many, but the one that's closest to my heart is Macropostrongyloides baylesi. It's a species of roundworm which the adult stage lives inside the large intestines of kangaroos and wallabies. And kangaroos and wallabies are normally just full of worms. It's like they're hopping around carrying entire ecosystems of worms in their stomach. Most of them don't cause any harm in healthy, free-ranging kangaroos, so technically, in my opinion, they're not bad parasites. Out of the several hundred species of roundworms that infect kangaroos, Macropostrongyloides baylesi is my favourite. I mean, from a distance, they're just small, plain, pinkish-white little worms that were thought to be a generalist species, meaning that they weren't picky about their hosts, would find them in eastern grey kangaroos, red kangaroos, wallaroos, all over Australia. But when we looked at them closely at their genetics and morphology, there were several different species, which were mostly host-specific. But the most fascinating thing about them was the detail of their morphology that hadn't been noticed before. Things like the patterns on their tail and the shape of their head. To me, Macropostrongyloides baylesi was a classic representation of the hidden diversity of parasites. They may appear small and insignificant, but when you look closely, there's so many things to be discovered, and that's pretty awesome.
Hello everyone, my name is James Sams and I'm a medical sciences graduate from Macquarie University, Sydney. I'd like to start by acknowledging that this recording is taking place on the traditional land of the Bidjigal people of the Darug Nation and pay my respect to elders past, present and future. So what is my favourite marsupial? I'd have to say it's the numbat, also known as the banded anteater. Native to Australia, this diurnal compact marsupial has a tongue that spans 1/3 of its body length, allowing it to burrow into crevices to obtain its mainly insectivorous termite diet. With rapidly deteriorating habitats and parasitic vulnerabilities, there are estimated, unfortunately, to be fewer than 1,000 numbats nationally, classing them as an endangered marsupial species. One very interesting but obviously problematic parasite for numbats is acanthocephalon, also known as spiny-headed worms due to their reversible proboscis. These parasitic worms shed very few eggs and are thus commonly difficult to diagnose until obstruction of the gut occurs in the host. Luckily, though, a broad spectrum of anti-parasitic agents are able to treat spiny-headed worms, and work is currently underway to treat captive numbats. Numbats are extremely cute, as you can see from the image above, but they also play an important role in the niche ecosystems they inhabit, in that they eat roughly 17,000 termites daily, greatly reducing the population of an overtly destructive insect species. And though I really appreciate looking at spiny-headed worms and their ability to turn their heads inside out, I'm happy to remove them in order to protect our Australian anteater. Thank you for your time.
G'day, my name is Dr. Sarah Preston and I live in Victoria in Australia. Thank you for asking me the question, what is your favourite marsupial parasite? Now, just for your future reference, you should never ask the parasitologist what their favourite parasite is. It's a question with too many answers. It's like asking a person what their favourite ice cream is. Is it caramel or is it honeycomb? Oh, hang on. Could be peppermint. The list goes on. Luckily, you have narrowed it down to a marsupial. The marsupial I've chosen to speak about with you today is the wombat. Now, you can see a wombat behind me. This is Nalia. And if I move slightly, you might be able to see her joey, Honeybun. Honeybun is quite rare because she was born as an albino joey from the wombat. These photos are from our wombats from our local wildlife park, and the credit would have to go to one of the a student of mine called Emily Flatters. Now, luckily, these wombats are in very good care, so there would not be, it would be very unlikely that they would have had exposure to a parasite infection, but if they were out in the wild, then they could be exposed to some nasty parasites, such as ticks and mites and some worms. One worm that the wombat in the wild is sometimes exposed to are worms that we call tapeworms. Now, tapeworms are called tapeworms because they basically look like a tape or a piece of tape, like masking tape. They're very long and flat. I always think they look like the pasta fettuccine, and I do like my fettuccine pasta. Members of this class of parasites are called endoparasites. That means they live inside the host, or they live inside the wombat. They're also flat worms, because when you take them out, they're very flat. They also lack a mouth and a gut. Now, tapeworms are very interesting because they have a head-like structure that we call a scolex, and they use this structure to latch onto the insides of their host. So in the wombat, they'd be living in the sort of small intestine part, so just before their stomach. And they've got this head-like structure called the scolex, but then after the scolex, they've got these long sort of segments, and these segments can grow and grow and grow, and they're very repetitive. And we also call these segments proglottids. And in these segments, they actually contain their reproductive organs. What's really interesting about tapeworms too is they contain both male and female reproductive organs, which means they could be a male or a female. They can pick and choose. Some of these tapeworms can be several meters long, and they can contain thousands of these proglottids or segments. The largest ever known tapeworm was actually found in a blue whale. So you can imagine how long that was. I think it was about 12 meters. There are some tapeworms that are quite small, so they only contain 4 or 5 segments or proglottids. So they can also be quite small. So most of the tapeworms that naturally infect wombats or marsupials don't cause much harm, but there is one nasty tapeworm that can be found in wombats sometimes. This is Echinococcus granulosus. It's thought to be introduced to Australia when the European settlers came, so when they brought over the sheep and the domestic dog. And what's happened is that wombats actually accidentally ingest these eggs, they ingest them from the environment, and these eggs will then turn into cysts, and these cysts sort of get lost in the wombat's organs, and they form cysts and then eventually cause damage, mainly through pathology, and they can end up dying with these cysts. For wombats, it's really common to see these in the lung area. However, in general, a wombat dying from these cysts or tapeworms that cause cysts is quite rare. Well, that's all from Australia, folks. I hope you guys can visit soon.
Hi everybody, my name is Scott Carver and I'm a scientist at the University of Tasmania. I'm here to talk to you today about my absolute favourite Australian marsupial, which is called a wombat. And wombats are the largest burrowing herbivores in the world. They are a kind of bear-shaped marsupial that digs a burrow and lives in that underground and comes out usually to forage for about 2 to 4 hours a day on surrounding grass. There's 3 species of wombat in Australia, and the one that I study is called the bare-nosed, or also called the common wombat. And my favourite fact about wombats is that they produce these cube-shaped poos and that help them aggregate in areas, and they use these as a form of chemical communication. Some colleagues and I won a Ig Nobel Prize a few years ago for discovering how they did that. So that's one of my fun facts. But fun facts aside, I also want to talk about sarcoptic mange disease, which is a major disease of wombats. And it was introduced to Australia by European settlers and their domestic animals. And it's mostly an animal welfare issue of wombats. They're highly susceptible to this, and they develop crusted mange disease, which is driven by a type 4 hypersensitivity reaction. Mange disease in wombats leads to alopecia and hyperkeratosis and all of those other sort of typical symptoms associated with mange disease in many animals. And they lose heat to the environment, they have elevated metabolic rates, and generally suffer a slow decline or they die from it. It can occasionally cause population effects, such as local population declines, but mostly it causes individual deaths and it persists in their populations without sort of causing widespread local extinctions and those sorts of things. It can be treated, and we are working on various treatments for that. But it's tricky to do, and it's an area of our active research. So thank you all for listening to my talk.
Hi, my name is Nina, and I study medical sciences at Macquarie University. Today, I want to talk to you about my favourite marsupial, the quokka. The quokka is Australia's cutest marsupial, found only in Western Australia, mainly on Rottnest and Bald Islands. But sadly, the world's happiest mammal is highly susceptible to parasitic diseases due to introduced species carrying them to the islands. Toxoplasmosis is a protozoan disease and is just one example. And feral cats have brought Toxoplasmosis gondii to Australia and spread it to the islands in Western Australia's waters. There is evidence to suggest that this parasite has the ability to change the behaviour of the quokka, making it more susceptible to predation. This means that feral cats could more easily hunt the quokka, completing the life cycle of toxoplasmosis and ensuring that it finds its way back to its definitive host, the cat. Quokka's populations are declining, and the species have been labelled vulnerable by the International Union for Conservation of Nature. And while it doesn't appear that toxoplasmosis is a direct cause of this decline, it certainly could be cause for concern. Sadly, very little research has been done to determine whether this infection is a significant threat to quokkas. Studies are generally outdated, with only one study from 1966 really investigating the link. However, the data collected was merely an estimate, with poor sampling and control methods. A paper in 2016 looked at the limited available evidence and found that Toxoplasma gondii could be a major threat to quokka populations. More research needs to be done to prevent our quokka populations from becoming endangered or extinct. And this research really needs to incorporate better sampling and control measures in order to collect appropriate and reliable data. Unfortunately, providing treatment to our wild quokkas is an unlikely solution, and prevention methods are also difficult and expensive. And due to the limited research, it's hard to know to what extent we need to address toxoplasmosis in our quokka populations. In captive populations, however, enclosures are effective at preventing feral cats from accessing the animals, and some medications are also available and shown to be effective against the disease. I hope you've enjoyed hearing all about quokkas today. Help us protect these vulnerable critters.
Hi, I'm Ruth. I'm a student at Macquarie University in Sydney, Australia, studying a Bachelor of Medical Sciences. So my favourite marsupial is Sarcophilus harrisii, or harrisii, commonly known as the Tasmanian devil. I first got to know about it as a child through the cartoon Looney Tunes, where the Tasmanian devil named Taz was depicted as a vicious and ravenous beast. However, if you look at pictures of the Tasmanian devil, I think it's actually quite cute. It looks like a black baby bear. So the Tasmanian devil is the largest carnivorous marsupial in the world, found only on the island of Tasmania in Australia. They usually feed on small prey such as frogs, birds, fish, and insects. Tragically, the Tasmanian devil population plummeted because of a widespread illness called the devil facial tumor disease. The marsupial is currently listed as endangered by the International Union for the Conservation of Nature. So one parasite of the Tasmanian devil is Toxoplasma gondii, or T. gondii. It is a protozoan parasite that infects warm-blooded animals, including humans, and causes the disease toxoplasmosis. T. gondii has multiple hosts. So it has its definitive host where it can reproduce sexually, and these are cats or members of the feline family. The cat sheds T. gondii oocysts in its feces. Now, oocysts are egg-like structures that are highly resistant to damage. And the Tasmanian devil can get infected with T. gondii by ingesting these oocysts or any meat containing live parasites. The oocysts turn into an infectious form and localize in the muscle and neural tissues. We still don't know the full effects of a T. gondii infection in Tasmanian devils, so hopefully further research is done and I can keep admiring these cute animals.
Some great stories there about parasites in marsupials, and I'm going to follow on from Ruth and talk about other parasites in Tasmanian devils. So the Tasmanian devil is threatened by this transmissible cancer, Tasmanian devil or devil facial tumour disease. And basically in Australia there's a national recovery plan to recover numbers and protect the Tasmanian devil and stop them from going extinct. And really interestingly, as part of that plan, it also includes a defined goal to save the Tasmanian devil parasites. And the reason for this is that the Tasmanian devils have some very unique parasites that don't occur in other species. So if the Tasmanian devil goes extinct, so do some of these parasites. But before I go on, I'd just like to acknowledge Liana Waite here, who's also on this slide. And Liana is a former student, and a lot of the data that I'm presenting on Tasmanian devils was undertaken by her when she was studying with me, and we're continuing to look at parasites in Tasmanian devils today. So coming back to this elusive parasite, it's called Daisy-Uretina robusta. It's a form of tapeworm, and as I said, it's only found in Tasmanian devils. So if we lose the devil, we lose a parasite species. And given this, the Daisy-Uretina robusta is listed as a threatened species. Daisy-Uretina robusta is listed on the Tasmania's threatened species list and identified as rare. And for all we know, the parasite may actually be extinct. We do know of other parasites that are present in Tasmanian devils, and so there's 25 species of parasites that have been described and reported. So 7 species of nematodes or roundworms, 9 species of platyhelminths or tapeworms, 3 species of protozoa, and 6 species of ectoparasites, so ticks and lice as examples there. So Liana did some work looking at the distribution of, or the occurrence of the different parasites in Tasmanian devils throughout this captive breeding and recovery, or what we would term the Tasmanian devil insurance population. when captive in this insurance population, captive devils are bred and maintained under different conditions. So there's kind of intensive housing where you have a couple of devils together that might be a breeding pair or young offspring or juveniles, through to the next stage up where we have big open plan cages, but they're still enclosed, and then finally into animals that are in the wild. And you can see here depicted on the slide that we have wild intensive captive, so that's those small cages with a few animals, or smaller, they're not small, but smaller cages with a few animals, versus these free range large enclosures, but they're also in captivity there as well. So you can look at the graph, and the different parasites are along the bottom. So we've got a roundworm here, Baleascaris tasmaniensis, woolly-eye sarcophili, Anaplatina daisyiurii, another tapeworm, but it actually has a broad host range and is found in quolls, as well as Tasmanian devils. Then we get into the protozoa, some coccidia, which is a common term inclusive of the parasite that I mentioned earlier on in the introduction, Iberia, and then also Cryptosporidium here, and finally Giardia. So 3 protozoan parasites, my favourite parasites. And what these bars tell us, that if we look at animals in the wild, so the red bars, and on the prevalence, we can see that 25% of animals sampled from the wild had this parasite, okay? Whereas if we come across this Cryptosporidium, almost 50% of animals in the wild were carrying the Cryptosporidium parasite. And so we can really just see here that, Tasmanian devils have lots of diverse parasites, or these particular groups of parasites, and they're still present within the insurance population, which is really important because parasites are an important part of the ecosystem and their host. And if we're managing animals in captivity and removing their parasites, then we're upsetting the balance of that relationship between parasites and their hosts. And who knows, perhaps we're possibly driving parasites to extinction. So it's interesting just to note, while we're talking about parasites, and this slide gives me the opportunity to say, most of the time animals are infected with more than one parasite. So we don't have one species, we don't have one worm or one protozoa. I mean, humans tend to be like that in some areas, but when we're talking about wildlife and in their natural conditions, they're usually carrying more multiple parasite species. And that's what this little graph here shows. So we can see here that if you look at basically the bars, there's 4 colours, white, pink, and like a reddy dark red there, and then black. And this is suggesting whether they're within the animals sampled, whether they're carrying 0, 1, 2, or 3 parasites. And really, it's quite a detailed slide, but what I'd like to go away with is that, if you look at the different sites we sampled, or basically the different areas where we sampled Tasmanian devils, the animals in those areas are carrying multiple parasites, and as shown by these different coloured bars. So what do Tasmanian devil parasites look like? So here's some great photos that Liana took, and these parasite stages are eggs and what we would term oocysts for protozoa, and you actually purify these out of Tasmanian devil poo. So we collect the poo, take it to the lab, and then apply some techniques to remove the parasite stages. So you can see here a tapeworm egg for Anaplatina daisyiurii, and then a nematode or a roundworm egg here for Woolly-eye sarcophili, and then finally this unknown species of coccidia, or possibly an Iberiad. So my favourite parasites, the protozoa. But I'm going to talk about 2 of these today a little bit deeply. Actually, none of these today on this slide, but I'm going to talk about 2 further species a little bit more. And you've heard about one species called Cryptosporidium from Manushika today. And so Cryptosporidium is a protozoan parasite that's found in a diverse range of animals, including marsupials. So marsupials in Australia have their own species, which you can see on the slide, Cryptosporidium fayeri and Cryptosporidium macropodum. So these Cryptosporidium species are found in a wide array of marsupials and even have been identified in the odd case of humans. Okay, so we went out and were looking at Cryptosporidium again in Tasmanian devils and looking to see if Cryptosporidium was more common in either the captive breeding program population, so those animals that are being intensively bred and managed, or in those free-range enclosures, versus what we saw in the wild. And what's really interesting is we see that animals in the wild have more cases or are more likely to have Cryptosporidium. So 45% of animals, Tasmanian devils in the wild, we detected Cryptosporidium in 45% of animals that we sampled from the wild, and then in less than 10% in those captive populations. So this is really good news for the animals in captivity. You know, they don't have parasites, but it's also really good news for the animals in the wild because, you know, often Cryptosporidium, and particularly species that are host specific, don't cause disease or impact animals in any way. And I should probably just mention that and in some of the other sessions, they may have talked about host specificity. And I mentioned this with Daisy eurytenia, that tapeworm that's only found in devils, is that some species of parasites are very host specific and you can only find them, say, in certain animals, whereas others have a broad host range. And for Cryptosporidium, some species of Cryptosporidium are very host specific. So for example, those marsupial species, whereas others such as Cryptosporidium parvum can be found in a whole range of animals, including cows, bats, people, and so forth. Okay, so it's called host specificity. So we can also look at the DNA and compare our Cryptosporidium samples that we got from Tasmanian devils to other Cryptosporidium species. And we can put it in a little program, I guess, and ask the DNA strands to this program to compare the DNA strands from parasites from different animal species. And so what we've got on this slide is what we would term an evolutionary tree of Cryptosporidium from Tasmanian devils. And basically you can see that we found a couple of known species of Cryptosporidium in our devils in the study. So here, Cryptosporidium fayeri, which we've already detected in other marsupials. And on the slide, you can see it's the one at the top. And you can see that we found that in one wild devil and the little image of the kangaroo next to the slide, showing that this species is also present in kangaroos. If you look to the very bottom of the slide, we found two additional species, Cryptosporidium galli and Cryptosporidium urus. These species are common in rodents and birds, and rodents and birds are actually form diet components of Tasmanian devils. So it's not unlikely that we would find these there because they may just be passing through their food item and being detected in their poo. So not necessarily an infection. But what we did find was an entirely new species of Cryptosporidium in both wild and captive devils. And we found it in a good number, like 38 animals that we looked at were carrying this Cryptosporidium type. And so we still need to go on and determine other factors about this Cryptosporidium to describe it as a new parasite species from Tasmanian devils, and to find out whether it's in other species or only specifically found in Tasmanian devils. So another parasite that we looked at was Giardia, and we were here interested in asking the same question: is there a difference in carriage of Giardia between Tasmanian devils that are in the wild or in those captive situations? And what we can see, definitely, we have low numbers of parasites in animals that are in captivity versus the wild. And again, we can do that genetic study. And on the slide, you can see the tree again, but this is for Giardia. And then right down the bottom, you can see that similar to Cryptosporidium, we actually found new types of Giardia in Tasmanian devils as well, which was great for us. You know, we're finding more diversity and more parasites, and really understanding the types of parasites that are present in wildlife in Australia. But then right up the top there, you can see that we also found a Giardia type that typically occurs in humans. And it's another aspect of the research that we're doing is to try and understand what we would term reverse zoonoses, or the transmission of pathogens or parasites that are typically associated with humans to our wildlife. So we hear a lot about zoonoses, and particularly now, given COVID, so a virus that has emerged from a wildlife species. But here we can have the reverse, excuse me, where parasites of humans can get into our wildlife and may have an impact there as well. So that really brings me to the end of my talk. And so what our study has shown so far, we know we have this elusive parasite called Daisy uratina robusta, a tapeworm in Tasmanian devils. We're doing trying to develop some tests and using museum specimens to see if we can find a DNA signature for that parasite, and then we can go out and start screening our Tasmanian devils to see if we can find it in certain areas, find that DNA signature, and then we can kind of work up from the DNA instead of working from the parasite down to the DNA. And so it would be really great if we could make that happen. But we've been in lockdown, so we haven't really been working on our museum samples for a while. It's also interesting that our research has shown that we have new species of protozoan parasites, Cryptosporidium and Giardia, and further studies are required on those to determine whether they're specific to Tasmanian devils or found in other marsupials. And then who knows? I'm losing my voice, I apologise. Who knows? Maybe there might be more parasites in Tasmanian devils that we need to conserve to preserve the biodiversity of parasites around the world. Thank you.
Hi, my name is Godfrey, and today I'm going to be talking a bit about parasite biodiversity in marsupials. So marsupials are most well known for the fact they have pouches, and there's lots of them in Australia. But I think they should also be known for the immense parasite biodiversity they host too. So as an example, this is a swamp wallaby, and 23 species of nematode have been described from these wallabies. There's 1 species in the esophagus, 16 species in the stomach, and 8 species that occupy the gastrointestinal tract. And within the stomach alone, your average swamp wallaby has 20,000 worms, which is a huge abundance and diversity of parasites inside these marsupials. So marsupials could be considered little hopping biodiversity hotspots for parasites. Today I want to talk about the woylie, also known as the brush-tailed bettong, and they're a small marsupial. They were once widely distributed across the southern half of Australia, but after European arrival, their distributions became severely restricted after habitat loss, introduction of predators, and so on. Now they occur in the southwest of Western Australia, and back in the 1970s, their populations started to decline there. These declines were largely attributed to foxes, and so a large amount of work went into controlling foxes, and this was really successful and led to woylie populations rebounding, and it was so successful, in fact, they were taken off the endangered species list. But sadly, the story doesn't stop there. Woylies started to decline again back in the early 2000s. And wild predators such as foxes and cats are important in these declines, the magnitude of decline that was observed couldn't be explained by predators alone. And so disease was thought to maybe be involved in these declines. So at the time, a lot of research went into trying to find out what parasites and diseases woylies had. And so in this diagram here, this is essentially a list of the parasites that are known from woylies so far. And in total, there's 37 parasite species that are known to infect woylies. We have ectoparasites on the left and endoparasites on the right. And those that are highlighted in yellow are thought to be host-specific. And you'll notice among the ectoparasites, there's actually a tick, Ixodes woylii, which is, as far as we know, host specific. It only occurs on woylies and nothing else. And so they too are little biodiversity hotspots for parasites. Coming back to the decline, among the parasites that were found, one group in particular that was particularly interesting from the perspective of the decline, and these are trypanosomes. So trypanosomes are blood parasites. They live in the bloodstream of the woylies, and they are transmitted from one woylie to another through a vector. And for these trypanosomes, we think maybe ticks are involved. And trypanosomes are known to have pathogenic potential. So other members of this group of parasites cause sleeping sickness in humans. So at least 3 species, probably more, of trypanosomes infect woylies. And the 2 pictured here, Trypanosoma copemanni on the left and Vigrandis on the right, are the ones that most commonly infect woylies. Of these 2, Trypanosoma copemanni is particularly interesting, and that's because they've been found to be capable of infecting tissues, and pathological lesions in dead woylies look very similar to those that we see in humans infected with trypanosomes. So we were able to look at blood samples that were collected from a woylie population that was declining, and we looked at how trypanosome infection patterns varied as the woylie population declined. And we had a couple of key findings. So one is that Trypanosoma copemanni was far more common in this declining woylie population than Vigrandis. And for both parasites on their own, copemanni and Vigrandis, their infection levels declined as the woylie population declined. But the interesting thing was that even though our woylie population was declining, the proportion of hosts that had both of these parasites actually increased. So even though we're losing hosts from this system, the hosts that are remaining had a higher proportion of those hosts were infected with both copemanni and Vigrandis at the same time. And so this was a bit puzzling, but we've come up with one explanation for why we might observe that. And that is that maybe these parasites are interacting with each other. So take an example where copemanni is in a woylie on its own. And so it's replicating away and it's got nothing really to control it, which means that the more it replicates, the more negative impacts it has on its host. But if copemanni is in a host with Vigrandis, the other trypanosome, it suddenly can't replicate as much. It's got some competition within the host, and so that competition reduces the growth rates of copemanni and maybe Vigrandis as well, meaning that neither parasite can kind of grow out of control, and therefore by having 2 parasites in the same host, they might actually reduce the overall effect that those parasites have on their host. So this is just a hypothesis, but it would explain what we observed, where we see co-infections become more common in the woylie population as the woylie population declines. And I think it highlights the importance of thinking about the whole parasite biodiversity within a host and how those parasites might be interacting. So because the woylies are declining, they have been a focus of significant conservation management. And one of the key conservation management tools for woylies are translocations. So when we pick animals up and move them around, we are probably moving their parasites with them. But we know very little about how that changes host parasite communities and what parasites we might be losing or gaining through doing this. And so we were involved in 3 translocations where we moved woylies from a sanctuary into 3 different sites, and essentially we tracked their parasite communities after the translocation. And this work was largely done by a PhD student at the time, Amy Northover. And so what happened to their parasites? So firstly, let's take a look at our trypanosomes. On the left, we have Trypanosoma copemanni. This is the one we think is maybe pathogenic. And each of the different coloured lines shows a different site where the woylies were translocated. And what we see amongst this jumble of lines is that each site seems to be doing something quite different. So Dryandra, for instance, the black line, maintains a relatively high prevalence of copemanni, and the other 2 sites sort of jump around quite a lot. Vigrandis shows a similar thing where we see that each of the sites sort of do more or less their own thing. Each of the sites ends up with a different prevalence, so a different proportion of hosts that are infected with that parasite. And we see a similar pattern when we look at gastrointestinal parasites, and these are just a couple of examples. On the left we have strongyles, on the right we have coccidia, both gastrointestinal parasites. And the red arrow is the point at translocation, and then we have July and September, so after the translocation. And again, we see a similar pattern where the two sites this time that we're looking at, the parasites end up doing quite different things depending on the site that the hosts went into. And so it could be that there's different environmental conditions at those sites, different densities of hosts, maybe different alternative hosts within that community that might be shaping these host parasite communities. And ultimately what we see in these translocated woylie populations is quite different parasite communities emerging, and those communities become more similar to the resident woylies that are in those places. And so when we move woylies, we do change their host parasite communities, and we're looking at more research to understand exactly how they change and what parasites we might be losing, as well as which ones we're gaining. So I'd like to thank you all for listening, and I hope you enjoy the rest of Parasite Biodiversity Day.
Hi, Dr. Godfrey.
Hi, how's it going?
Good. Thank you for joining us for this Q&A session. And once again, for our audience, if you have any questions for Dr. Stephanie Godfrey, please feel free to put them in the chat of the YouTube. So our first question here from audience, Sandra Avila, asked, how do you handle the animals to take the parasite samples? Do you put them euthanasia or euthanasia?
Good question. So I guess we try not to if we can get away with it. And a lot of the parasites that we're looking at, we can measure from their poo, basically. So when we trap the animals, they usually enter the trap overnight. And then when we catch them the next day, there's usually a lot of poo in the trap. And so we can measure their gastrointestinal parasites that way. And for their blood parasites, we take a blood sample from the tail. So usually we can get that without having to put them under anesthesia. So yeah, good question.
Thank you. And the next question, was your discovery of both parasites being present in the woylies something you have seen in other hosts?
Oh, good question. So I guess, one of the challenges is we tend, when we're looking at parasites, sometimes we're sort of just look at one parasite and we tend to not look at the others. So co-infection is something that we don't think about as much as maybe we should. I think with the trypanosomes, there's been a lot of people working on them, and we're discovering more and more the diversity of blood parasites that occur in our marsupials in Australia. But yeah, it's something that we tend not to think about too much, but potentially is quite important. So yeah, good question.
Thank you. And we also have Dr. Michelle Power joining us. Hi, Michelle.
Hello, everyone. How are we doing? Hello, Stephanie.
Hi, Michelle.
We have a question from Michelle as well. A question from Sandra. It's about Toxoplasma gondii. It's a marsupial capable of exerting oocysts. If so, are they infective?
So the marsupials don't produce the oocysts, okay? Only cats can produce the oocysts of Toxoplasma. So cats are what we term the definitive host, where those infective stages are produced. Marsupials are kind of accidental hosts. So yeah.
Thank you. And a question for both presenters. Is host specificity of Cryptosporidium or the Exodus host dependent, so host specific immunity, Or do they think it's more associated with spatial or environmental constraints? Who wants to go first?
Sorry. Was that host specificity of Cryptosporidium?
Yes.
Is it associated with the host or the environment? Was that the question?
I also put it in the chat here.
Yeah, okay. Okay, so I can talk to Cryptosporidium. No, it's definitely associated with the host, okay? So the Cryptosporidium has marsupial-specific species that are only found in marsupials. There are some reports of those species found in people, so zoonoses or transmission from animals to humans. But also, there is some environmental constraint because marsupials are mainly in Australia. And so for what we know, those species are present in Australian marsupials, but one of them has been found in the opossum in North America.
And I guess to add to that, the ticks, the exodes that are host specific to the woylie, I guess we don't really know why they're host specific, but one of our thoughts is maybe to do with the behavior of the woylie and its nesting habitat is quite specific to any of the other marsupials that live in those environments. So that's going to influence the way that tick can get from one host to the other. But it's possible it's something to do with the host as well, and maybe that tick has specialized in some way that it can persist on the woylie. So again, I guess similar to Cryptosporidium in that it's probably a bit of both environment and behavior and ecology as well as the parasite adapting to its host.
Thank you. And a question from Dr. Chorsk asks our both presenters, do marsupials differ from placental mammals in their immune response? For example, leukocyte cells involved, cytokines, timeline of responses, and so on. Who wants to go first?
Do you have anything to say, Steph?
I have nothing. I have no idea, Michelle.
Okay, yeah, look, I don't have much to say either. I'm not an immunologist, I'm a parasitologist. I do know that what I can say is I do know that there's a lot of information out there on the production of antimicrobial peptides in the pouches of marsupials to protect the juveniles in those early stages of life. Look, my memory's catching at me to say there is some variation in the immune response, but I think it's minor tweaks here or there. I still think it's pretty standard innate and active immune responses like in placentals. So sorry, I can't quite answer that either.
Anything you want to add, Dr. Godfrey?
No, I think Michelle did a great job. I have no idea.
So the next question is to you, Stephanie. It's from Sarah Valeria. Asked, do you know the prevalence of parasite coinfection in woylies?
Good question. So I'm just trying to think back to my presentation. I mean, in terms of the trypanosomes, I think the rates of co-infection were certainly not super high. It was probably somewhere around the either 20 to 40%. And in woylies in general, I mean, we know that woylies are infected by more than one parasite. So, they've got trypanosomes, but they've got gastrointestinal parasites as well as ectoparasites. And so most woylies would be carrying more than one of those things. So I would certainly say it's probably a large majority would be carrying multiple parasites. Yeah.
Thank you. And for our last question is to Michelle from Sandra. If you find an unknown coccidia, what's the next step? Do you make a molecular analysis?
Yeah, exactly. So I mentioned in the talk that we did identify some coccidia that we think are unknown. There's no previous reports of coccidia in Tasmanian devils. And so we you saw there was a photo of an oocyst there, so we can do morphology on that. But then we would purify those out of the sample and do the molecular test to work out the sequence, which would then tell us what genus it belonged to, whether it's a new species or not. So that would exactly be the path that we would take to try and uncover a mysterious parasite like that.
Thank you, Dr. Power and Dr. Godfrey again for the presentation and answering questions.
Thank you very much.
Thanks very much.
So in the interest of time, this is the end of our parasite and host animals with pouches session. And we'll take a 3 minutes break before continue to our next session, Parasites and Arts, with Dr. William Campbell's interview and the Maguro Parasitological Museum in Japan. See you soon.